Now we will talk about some of the molecular ways to figure out if and how much gene flow is happening between populations, and specifically in this video we're going to talk about quantifying gene flow of discrete populations, meaning we already have some kind of preconceived idea about which population, what the population boundaries are, and which individuals are in those populations. So one of the main ways that is used to estimate genetic differentiation between populations is called FST, or the fixation index. So in this image here, again, this is dependent on the fact that we already know what population A is and what population B is, and then we are able to calculate some kind of FST metric to compare those two populations to see whether gene flow is happening and how different those two predetermined populations are from each other. In terms of what do these values look like and what do they mean, a higher FST value indicates more genetic differentiation. So FST values are on a scale from 0 to 1. The higher that value is, the more genetic differentiation there is. There are some examples in your textbook about how to calculate FST. We're not going to focus on that because, similar to when we talked about genetic diversity in the last module, I've never had to know how to actually calculate FST. There are always analysis programs that do it for you. So we're going to focus more on the interpretation of it rather than how to actually calculate it. So as I said, the higher the value, the more genetically differentiated those populations are. So let's talk a little bit more about both ends of that spectrum. If two populations have the exact same identical allele frequencies, that means they are not genetically differentiated and their FST value would be 0. So an FST value of 0 means two populations are genetically identical. So if we look at this example from scarlet tiger moths, this in the table is showing two different populations, population 1, population 2, and it is showing the genotype frequencies for the different possibilities shown here. If you look at the genotype frequencies across those two populations, they're all relatively similar. And if you actually calculate FST for these populations, you get a value of 0.009. So not quite 0, but very small. So this indicates those two populations are very similar to each other. In contrast, if two populations are fixed for different alleles, that means you only find one allele in one population and you only find the other allele in another population, that gives you an FST of 1, which means the populations are very genetically different from each other. So in this example here, these are again those scarlet tiger moths, and this is showing the different possible phenotypes that they can have. So some have really heavy white spotting on their wings. Some of them don't have any white spotting. So let's assume that if you have heavy white spotting, that means that only the capital A allele is present. If you have no white spotting, that means only the lowercase a allele is present. If this is the only allele present in population 1, and this is the only allele present in population 2, if you calculate FST for those two populations, FST would be 1 because they're completely genetically different. They don't share any alleles at that site. So those are the extremes in quote-unquote real-life scenarios. You'll basically never get those extremes. What you'll get is some kind of FST value somewhere in the middle. So this table is showing some very rough guidelines for how to interpret levels of FST numbers, values that you would get, and how you would interpret them. So again, very rough estimates, but in general, an FST value between 0 and 0.05 indicates there is little genetic differentiation. Between 0.05 and 0.25 means there's moderate genetic differentiation, and above 0.25, that's where we would say there is strong genetic differentiation. Again, there's a huge caveat here because the FST values that you get when you're calculating this with real data sets is very dependent on what type of species you're looking at. So in marine species, for instance, the scale is often adjusted because there's often a lot of dispersal in marine species because there are not big barriers like mountain ranges or anything like that. And so for marine species, for instance, we don't often see levels of differentiation that high in any species. So again, big caveats here, but these are some general guidelines in terms of where we would start to see cutoffs for strong versus weak genetic differentiation between populations. And one thing that's really cool about calculating FST values now is that we can look at FST values for a ton of different loci, especially if we're using high throughput sequencing approaches like RADseq. So this is some data from my own work. This paper I've referenced several times now throughout different lectures and even in lab. This graph here is showing a FST distribution, which is a histogram. So on the x-axis is the FST value. So again, in this data set, what we're doing is comparing northern versus Southern California snails. And so this FST estimate is comparing those two different populations to each other. The y-axis here is the frequency. So that means for how many SNPs did we find that actually had that calculated FST value. So overall in this data that was calculated from SNPs that were identified from RADseq, there were roughly 2,300 total SNPs in the data set. This frequency histogram is showing for all 2,000 of those SNPs roughly what were the FST estimates for them. So what this histogram or FST distribution is showing us is that overall, most of the loci are very genetically similar to each other. Overall, the average FST for all 2,000 of these SNPs is only 0.0042. So it's really, really small. And if you look here at the peak of our histogram, the highest frequency of SNPs is centered right there around 0. However, if you look at the scale for this x-axis here, you can see it actually reaches all the way out to 0.6. And if you squint really, really hard, there's a tiny little bar right here. So even though the vast majority of those 2,000 SNPs have very small FST values, there are actually a few SNPs there that have high FST values. And if we think back to our neutral versus outlier adaptive loci, these loci are outliers. So for instance, when we looked at a couple of these SNPs in the lab that we did previously, we identified a few loci that were out of Hardy-Weinberg equilibrium, and those are the same loci we'll talk about later that through different methods you can confirm that they are under selection. But this frequency histogram for FST is indicating to us that we do have some FST outliers. Those are probably really interesting for us to look at because their differentiation between those two populations is so much higher than the average for the genome. So that gives us some indication that there might be something really interesting going on there, and the function for that particular gene that the outlier is in might be related to some kind of adaptation between the two populations. So when we're talking about FST values, like I said, the way that it's often done now in this realm of high throughput sequencing is you can calculate average FSTs for all of these thousands of loci that you're getting, but it's also really informative to create these distributions to see, okay, on average, yeah, most of our SNPs have very small FST values, but some of them are these outliers that have really high FST values, and so those are really interesting to investigate further. In addition to FST, one other way you can try to quantify gene flow molecularly between populations is to use something called assignment tests. What assignment tests do are assign individuals to their population of origin by comparing individual genotypes to the genetic profiles of multiple populations. So let's walk through this with an example that's shown here from tigers in a preserve in India. So say you have this one male here and you're trying to figure out which population he came from. What you would do is figure out his genotype, his allele frequencies. So that's the first thing you would need to do. The second thing you would need to do is obtain a genetic signature, overall allele frequencies from each of these possible populations present within that reserve. So then what you would do is compare the genotype you obtained from this individual to each of the possible population genotypes and see which one that individual male tiger matches the best. If you find that this guy's genetic signature most closely matches the MTR population, but you found him and physically sampled him in this other population, that would indicate that he is a migrant and he has moved to this other population. So that would be an indication that there is some gene flow going on. Again, you would have to monitor and see if he's actually reproducing. But this is another way that assignment tests can help us figure out, based on individuals, whether gene flow is happening between different populations.